I have changed the slide. Can you see it?
Yes, ma'am.
Okay. So it is responsible for mainly for the cervical cancer. Okay, so almost 50% of the cervical cancer, it is seen that the causative agent is human papilloma virus. So besides human papilloma virus, the cervical cancer, it can also cause cancer of the vulva, vagina in the females. Also, the anal canal, oral, and the penile carcinoma are also implicated. The human papillomavirus is implicated in all these different types, but in different proportions. So what is the virus like? It belongs to the Papilloma viridae. It can cause infection in different vertebrate species. More than 250 different types of vertebrate species can be infected by human papilloma virus. So basically, the virus has been co-evolving with the vertebrates. Okay. The important thing about the pathogenesis is they exclusively infect the epithelial cells in the epidermis and in the mucosae. So it does not go beyond that. It does not go deeper than that. Okay, so it affects the epithelial cells of the epidermis or the mucosae. So there is no viremia here. Almost more than 206 different types of human papilloma virus genotypes have been described, and of which around 18 different types, the genotypes, are associated with the cancer. So different genotypes are responsible for different types of cancer. Approximately 99% of cervical squamous cell carcinoma is caused by the one of the types, it's HPV 18. Some others are responsible for oropharyngeal carcinoma, others are responsible for anogenital, and these are called the high risk types, the ones which call cancer, and the others, the low risk or the benign types, they are associated with warts or condyloma acuminata. The virus, it is a non-enveloped virus, and it has two capsomers which surround 72 capsomers which surround the genome. The genome codes for different proteins, the early proteins and the late proteins. The early proteins, which are denoted by E, and the late proteins, which are denoted by L. So basically here, why I have projected this is there are two important genes which code for the different proteins, the E6 and E7. Okay, these are the ones which are responsible or which have the oncogenic property. These are the ones which are responsible for the suppression of the P53 or the retinoblastoma gene. It binds to these P53 or the PR. This R is the retinoblastoma gene, and it can cause the carcinomatous changes. This one, the minor capsid protein and major capsid protein, these have been incorporated in the vaccine, which is now available for this human papilloma virus. So the vaccine component is from this portion, the L. All these pictures are basically to tell you the different functions of the different regions of the genome. As we have said, different parts of the genome have different functions. Like E1 will have the function of will be responsible for viral replication. E2, which will regulate the transcription and replication. Similarly, the important ones are the E6 and E7, where the E6 it produces its codes for the oncoprotein, which binds to the tumor suppressor protein P53, and E7 again it codes for oncoprotein, which binds to the tumor suppressor protein retinoblastoma. These are the capsid proteins. The importance of this in the modern times is that these have been incorporated in the vaccine for the human papilloma virus. So as we have said, there are more than 206 different genotypes, right? And of which we can classify them as the high-risk genotypes. High risk because they are able to cause cancer of different sites, especially the cervical carcinoma. So 16 and 18, they are the ones which are most often associated with carcinoma of the cervix. Whereas these ones, 6, 11, 40, 43, these are the ones which are responsible for the warts, the different warts appearing at different sites on the body. Okay, so and some of the vaccines, they have all the vaccines have these genotypes which are responsible for causing CA cervix, whereas one or two of the vaccines, they also have the genotypes which cause the condyloma acuminata. So they can prevent the genital warts as well as cervical cancer. So you are supposed to learn a few names, few of the numbers which are very frequently associated with cervical cancer and very frequently associated with condyloma acuminata. These are okay. So how does the human papilloma virus, they cause the cancer? As we've seen, there are two viral oncoproteins of high risks, the E6 and E7. They promote the tumor progression by inactivating the P53 or the RB, the retinoblastoma suppressor gene. Okay, and the viral oncoproteins are capable of transforming the primary human keratinocytes from either the genital or the upper respiratory tract epithelia and disrupting the cell cycle regulatory pathways in the genetic progression of the squamous cell carcinoma. So basically, like all the other oncogenes, it will be disrupting the cell cycle regulatory pathway. There is an uncontrolled growth. That's why there is the CA, carcinomatous change. So as I said, these two are the ones which have the mucosal high risk, 16 and 18, responsible for around 50% of the squamous cell carcinoma of the cervix, around 35% of the cervical adenocarcinomas, and 25% of the oropharyngeal cancer, this HPV 16. HPV 18, again, it accounts for around 20% of the cervical squamous cell carcinoma, around 35% of the adenocarcinoma, and around 1 to 3% of the oropharyngeal cancer. So all the different HPV vaccines will have HPV 16 and HPV 18. Besides, the low risk ones like the HPV 31, 35, 33, these ones, they are also responsible for around 30%. All these are responsible for around 30% of the cervical squamous cell carcinoma. The 6, 11, 13, 22, these are benign genital lesions, the warts. They can be respiratory papillomatosis, some outgrowth of the skin tags that are seen in the respiratory, the mucosal tags that are seen in the respiratory tract. Okay, so these are the ones which have low risk or the benign ones, and the upper ones are the ones which are the high risk for CA cervix. He won the Nobel Prize for the association to proving the association of HPV with cervical CA cervix in 2008. So this one shows that almost 90%, more than 90% of the cervical carcinoma is where the HPV was positive. So it is a positive association of the virus with the cervical carcinoma. So basically, you have to understand that the human papilloma virus, it has a very efficient transmission cycle. As we have said, the keratinized cells from the skin They contain the virus particles, and if someone gets those skin or the shed skin or the viruses from that part, the other person can get infected. Okay. Second important property of this virus, how uh, it is a very efficient spreader, is that it can remain there in a latent stage and persist for a long time. And again, as the basal cell layer grows up in the epidermis, the virus particles also they grow within those cells and they are carried to the superficial layer. Third, they are restricted to the, as we have seen, to the epidermis and the epithelial cells of the superficial mucosa. Then, because of their restricted infection on the superficial sites, they are protected from the immune response. Okay, so it stays in a low profile. It does not go into the where the immune system are active, that is in the bloodstream, and that's how they can escape the immune response. And they can remain asymptomatic for long periods of time, and suddenly they reactivate and then cause infection. This one shows how the virus can, in long uh, duration of the infection, it can change from a benign lesion, asymptomatic lesion, to carcinoma cervix. Okay, this is the progression. So basically, this is the epidermis where the viruses are, these are the infectious viral particles. From the basal layer, it can come up and cause changes in the epithelial cells. They are shed from the superficial cells. After it takes many months for the changes to occur, okay, and many years for it to become a completely invasive CA cervix. So this is the transformation which has occurred in the cervical epithelium. So this is the transformation zone. This is by per speculum examination. This is where the histopathology, where there is the transformation of the epithelial cell. Same thing, normally the cervix appears like this. In low grade epithelial hyperplasia, it's like this. High grade carcinomatous changes. Same thing, low grade, normal, low grade, high grade cancer. So basically it takes a long time for the virus to cause the cancerous changes. This is the prevalence of cervical cancer. The areas which are in red, these have the higher prevalence. This one shows that both in male and females, the HPV are responsible for many HPV related diseases like genital warts, cervical cancer. The blue is the male. See, in the male, it's 17 million. Okay, 14 million genital warts in females. This is worldwide data. Cervical cancer, then head and neck cancer, anal cancer, then vulvar and vaginal cancer, and penile cancer. So these are the different. This is the spectrum of disease that HPV can cause. So how do you prevent all these? We have the HPV vaccination, which is now given in the for the adolescents before the sexual activity occurs. So HPV vaccination, it will reduce the HPV prevalence significantly, and in turn, it can prevent the cervical cancer as well. So these are the FDA approved HPV, the human papilloma virus vaccines. As I've said, the earlier ones, they just had the two high risk genotypes, HPV 16 and 18. These were given to the females to prevent specifically the CA cervix. The later ones, they are one that is goes by the name Gardasil is quadrivalent. It has 4 different genotypes, 6 and 11 to prevent genital warts, and 16 and 18 to prevent the CA cervix. It can be given to both males and females. The age group, as I've said, before they embark on sexual activity, they should be protected. This age depends on different countries. In the Western, they give it in early adolescence, and so it depends on the different sexual habits at different places. Gardasil 9, which is 9-valent, it contains some more of those genotypes which are responsible to cause the condyloma acuminata as well as the oral and the upper respiratory tract CA. So these can be given to both the males and females. So how do you diagnose the human papillomavirus? The virus cannot be cultivated. So the main diagnosis is by the clinical examination. Okay. And if you want to see whether if it is a cervical lesion, then if you want to know whether the cervical lesion has progressed to CA cervix, then we have to do a cytology by taking a pap smear. Then we can also have different biomarkers. As we've seen, we can detect the E6 and E7 genes. We can also look for those late the genome which goes for the late protein, the L1. Okay, by using the hybridization of PCR assays, we can either look for these or the early proteins E6, E7, the mRNA. To summarize, the human papillomavirus has there all the different vertebrae. It is ubiquitous. Also, they are associated with benign warts. Although they are associated with benign warts, they are also associated with carcinomatous changes in different sites of the body, especially the reproductive tract, external genitalia, oral, and pharyngeal. Males and females have different epidemiology and disease burden, and we have the vaccine to prevent these high risk genotypes from causing these carcinomatous changes. Okay, so basically for human papillomavirus, what I suggest is you should remember a few of those genotypes which are responsible for the different types of lesions. You should know the spectrum of diseases which are associated with HPV. And briefly, you should know how to diagnose the virus. The second virus that we are going to talk about, which is responsible for the STIs, the sexually transmitted infection, is the herpes simplex virus. Okay, we read about this virus when we were dealing with the skin infections. So just to recall what we have read, the herpes simplex viruses, they occur universally. Okay. Again, they cause intraepithelial vesicle from which the viruses are shed. They are transmitted from those lesions, or if it is herpes simplex virus type 1, they occur in the areas above the waist, usually in and around the mouth. We call them as the herpes gingivalis or stomatitis. Okay, so many of you must have experienced those lesions appearing around your mouth, especially during the exam times whenever there is stress. So from the lesions, the viruses can be shed, and from it can be transmitted to another person. During the primary infection, the virus enters the sensory nerve endings in the lesion and they are transported to the dorsal root ganglion where they remain latent. Later on, they reactivate and spread down along the same sensory nerves and again cause similar kind of lesions. This is true for all the herpes viruses. Okay, so if you recall what we read in varicella zoster, there is the lesion on the skin. They travel along the sensory nerves. They reach the dorsal root ganglion. Again, when reactivated, they come back along the same sensory nerves and then appear as secondary lesions. So same thing occurs in all the herpes viruses. True for herpes simplex virus also. Then they can be readily isolated from saliva or conjunctival fluid wherever the lesions are. Okay, also the genital ulcers. The herpes simplex virus type 2, basically mostly type 2, but nowadays it can be type 1 or type 2 both. They are responsible for the genital ulcers, and those ulcers are very painful. Okay, you might be asked, like in the genital ulcers, which ulcers are painful or which are non painless. So this one is the ulcers caused by the herpes simplex virus is painful. If you read syphilis, again there is the ulcers in the genital area, the external genitalia, but those ulcers that you see in syphilis, those are painless. Okay, so you keep track of the different organisms that cause external genital ulcers and make a note which one of them cause painful and which one of them cause painless ulcers. So this one, herpes simplex virus, cause painful genital ulcers. Okay, so these viruses can be grown in the different cell lines, the human embryo cell lines, and they can be the sample could be any of those lesions where the viruses are shed. Then how do you identify which virus has grown in the cell line? You use the immunofluorescence. So there will be the monoclonal antibodies which are tagged by fluorescent dye. You can use those monoclonal antibodies over the cell line which has the virus growing on them and then observe it under the fluorescent microscope. Or you can detect the DNA by doing the PCR. For herpes simplex virus, we give the antiviral drug acyclovir. Just to show you the different lesions that occur in herpes uh, infection of different parts. These are the vesicular rashes which later burst and then become ulcers in the genital area. It's usually the herpes simplex virus 2. Herpes simplex virus 1, this is the one that I was referring to, herpes labialis or stomatitis, if it is occurring at the edges. So basically the herpes viridae, these are the herpes, all the herpes viruses are double-stranded DNA viruses, right? So these are the spherical enveloped linear double-stranded viruses. The DNA viruses all replicate within the nucleus, and they in the lesions, if you take the scrapings from the lesion, base of the lesion, and stain it and see, you'll see intranuclear inclusions. The intranuclear inclusions are the replicating viruses in the nucleus. These are fast growing, they are cytolytic, and they remain latent in the sensory nerve ganglia that we have seen. And also we've also discussed that they reactivate when there is stress. They reactivate, they replicate at the site and then travel, causing the recurrent viral shedding occurs. Okay, so human herpes virus 1, 2, all these are different types of human herpes viruses. What we are concerned with are the herpes simplex virus 1 and herpes simplex virus 2, which are responsible for the sexually transmitted infections. So basically, how will you diagnose it is you can take any of the specimens wherever the infection has occurred. Okay, so for us in the sexually transmitted infection, this will be the relevant sample. The lesion swab is you'll take the swab from the base of the ulcer and you detect the antigen by immunofluorescence, or you can isolate the virus in those cell lines, or you can stain them, stain the host cells, and you look for the inclusion bodies, or the cytopathic effect that has been produced by the virus. Okay, so how do the stain smear occur? These the smears look. This is nothing but a Giemsa smear. We call it Zank smear from the ulcer scraping. And what is pathognomic is you will see the multinucleated giant cells. Within one cell, there will be multiple nuclei. Okay, this is known as the multinucleated giant cells. This is seen direct from the direct sample from the lesion. We perform the Zank smear and we look for the multinucleated giant cells. And this is true for all herpes viruses, not just for herpes simplex virus, but it can be for other herpes viridae group of viruses. This one is the antigen detection by immunofluorescence. If the cells, the host cells, have antigen, viral antigen presenting on the outer surface, if you label the monoclonal antibodies against those antigens with fluorescent dye and observe it under the fluorescent microscope, you can see these cells which will fluoresce. Okay, this is the swollen and rounded cells, which is the typical picture that is seen in case of the herpes virus infected cells. That is the cytopathic effect seen in the herpes virus infected cells. So that is basically the herpes simplex virus. So you should remember there are two different types, herpes simplex virus type 1 and type 2. Type 2 basically causes lesion below the waist, but now it is interchangeable. Type 1, it is because type 1 causes infection above the waist. The lesions appear as vesicles, which later burst and cause ulcers. And the ulcers are painful. The virus is a DNA virus. It can remain latent and again reappear. So the herpes virus lesion, they can recur. Okay, so there is a recurrent formation of ulcers. It will get cured by itself and then again appear sometimes later. That is because of the reactivation and replication of the virus. So that's the last of the slides. So basically, we have read about two different viruses today, which cause the uh, STIs. The herpes simplex virus, which cause ulcers, and Hello.